Welcome everyone to the July edition of Science and Nature Untapped, formerly known as Science and Nature on Tap, but now we are online. I will give everyone a couple minutes to settle in and we will get started shortly. Okay, it's 7:01. We will get started. So tonight we'll be speaking about Brian Bailey and the bats, the biological effects of mercury in bats. Before we get started, I'll just go over a couple of things. So for those of you, if this is your first time or if you've had some trouble in the past getting logged in, make sure that in the bottom right-hand corner that your volume is turned on. So if you can't hear me, your volume is off, bottom right corner on a PC. And secondly, if things are slow or if they're choppy, make sure that you have your windows closed at the bottom. You don't have extra applications running, and then up at the top, your browser window as well. You can tidy that up and close everything you don't need, and that should help your computer be happy and high functioning during this presentation. On the right-hand panel, there's a little kind of double one on top of another text message type of icon, and that's the live event Q&A. So in the right-hand side, you can probably see that I've already left 2 links, and we had Lee say hello, and then I asked for people to let us know who you are and where you're from. We'd love to know who we're speaking with. You can identify as your name when you signed in. You probably had the option, or you could sign in as anonymous. It's up to you if you want to put your name in there or not. And you can ask your question in the right-hand side there. It will be visible to everyone else. Today we're joined by Your TV. Gabe is connecting us with that. Thank you, Gabe. So we will start our Your TV coverage in 3, 2, and 1. Welcome to July Science and Nature Untapped. Brian Bailey and the bats, the biological effects of mercury in bats. We begin this event by acknowledging that many of us are living on unceded land and traditional territory of Indigenous people. We're grateful for the opportunity to live where we do, and we thank all the generations of people who have continued their responsibilities to Mother Earth since time immemorial. I would love it if everyone would follow us on social media. So we are on Facebook at River Institute, at St. Lawrence River Institute on Facebook is our handle. On Instagram, it's at River Institute C. It's also at River Institute C on Twitter. If you'd like to sign up for a newsletter, you can go to our website, riverinstitute.ca, and at the bottom of the page, you can sign up for that. Upcoming River Institute events. So the Ontario government did allow summer camps with specific rules and regulations to be followed, but given those rules and regulations, we were allowed to open up summer camps. So EcoFriends is canceled this year, but we are going to be running Junior Scientist Camp. And there's a couple spots available for next week. Junior Scientist Camp is for ages 12 to 14. Additionally, we're doing a call out to nominate someone for the River Award. The River Award was established in 2009 to coincide with the River Institute's 15th anniversary. The purpose of the award is to recognize and encourage environmental stewardship and responsibility in the community. The recipient of the River Award is an individual, organization, or company that exemplifies one or more of the following. Sets an example of environmental awareness, conservation, and respect for the St. Lawrence River and or associated watersheds in his or her daily life, work, and or community activities. Provides a significant contribution and or innovative approach to a sustainable river ecosystem. Encourages others to be good environmental stewards. In the Q&A on the right-hand side of your screen, for those of us who are online, I have included the CAMPS website as well as the River Award nomination form. For those of you watching on your TV, you can get to the CAMPS registration or more information at riverinstitute.ca/camps. To nominate someone for the River Award, it's riverinstitute.ca/river-award-nomination-form. And again, both of those links will be posted on our social media. So please follow us and you will hear all the great stuff that we're doing. August 5th, September 2nd, October 7th are our next Science and Nature Untapped. We'll continue to do these online. And then October 28th and 29th is our River Symposium. So stay tuned for more details on that. That's everything from me. So I will hand it over to Brian, Dr. Brian Hickey. I have unshared my screen, so you are welcome to share your screen now, and I will pass it over to you. And don't forget to unmute yourself.
All right, well, thanks, Katie. It's a great pleasure to have this opportunity to talk to everyone who's watching and listening about the recent research Bailey and I have been doing. An alternate title for this talk could also be Mercury, Why Are Bats So Different from Other Vertebrates and especially other mammals? So I'm going to start by giving you a bit of an overview of what I'm going to talk about and what Bailey as well is going to talk about. I'm going to start with a little bit of an overview of bats of Ontario and also the threats to bats in North America in general and in Ontario in particular. I'm going to talk a little bit about mercury and why mercury is of interest to someone studying bats. I'm going to talk about why bats, or I'm going to show you that bats behave very differently compared to all other vertebrates when it comes to mercury. And then finally, we'll talk a little bit about the impacts of bats on mercury. At that point, I'll switch over to Bailey Bedard, who's doing some really interesting work about how mercury affects bats at the molecular level, at the level of the molecule and the level of the gene. So many of you probably know that bats recently have declined, and many species, including some of the species we have here in Ontario, have declined by more than 90%. And a lot of people have heard that, but I don't think it really sinks in what that really means, a 90% decline. We could be witnessing before our eyes the largest crash of any mammalian population since biologists have been measuring those sorts of things. And they say a picture is worth 1,000 words. And here's a picture of a cave that I visited in 2009. And in every year back in that day, I would visit 3 or 4 different hibernation sites as part of routine monitoring. And the pictures you see here in this slide, that's what I would see every time I would enter a cave in 2009. This is what I saw in 2016. And this isn't just one cave or one abandoned mine. These are all of the sites that I visited, all of the sites that I had previously used as research sites. So a picture's worth 1,000 words. That's a real dramatic change. 2009 and 2016, really not very a very long time span. And most of the reasons for this dramatic decline are well known to some of you. It's due to the spread of an invasive exotic fungus, a fungal disease that causes white nose syndrome. And if you look at the cursor here, this is where it was first detected in the winter hibernation season of 2006, 2007. And then a year later, it spread into the blue area here. And then by 2014-15, it was in the brighter yellow. And finally, by 2019, it's the red areas here. So it spread rapidly. It started in Upper New York State in 2006, 2007, and about 10 years later, it reached the far east and west coasts, and it's still continuing to spread. And so that's the most well-publicized reason for bat declines, but that's only one of several threats bats face. Bats face threats due to massive scale wind turbine development. They face threats, and I'm just going to move here from the wind turbine on the left-hand side. I'm going to move counterclockwise through these images. They face threats due to land clearing, sometimes clearing of old buildings they use for roofs, sometimes land clearing forests that would have been places where they would find abundant prey. Solar farms take up land, urban development takes up land, farming that's moving from small-scale family farming to industrial size scale farms is taking up land. And of course, bats in Ontario, all bats in Ontario, and I'll talk about the species in Ontario in a minute, all the species of bats in Ontario eat insects. Insects also are often pests of our forest and agricultural crops, so they're the target of insecticide spraying. Insecticide spraying hits bats in two ways. First of all, bats can be inadvertently contaminated by the chemical pesticides, but also the insects that are the targets of the spray obviously decline in population numbers, and therefore the bats' prey, the bats' food also declines in numbers. So in Ontario, we have only 8 species. Bats as a whole make up almost a quarter the species of mammals on the planet. A little over 20% of mammal species in the world are bats. There are more than 1,300 different species worldwide. We only have 8 in Ontario, and I've shown images of 5 of them here. And different species are subject to different individual threats. So if we start at the bottom left, the little brown bat, that's the species that's most heavily impacted by white nose fungus. In addition to little brown bats, the 3 that I've listed just in a column on the right, the small-footed bat, the tricolored bat, and the northern long-eared bat, they're also heavily impacted by white nose syndrome, white nose fungus. And the reason for that is those are the species that hibernate in caves. As I said, all bats feed on insects, and insects are not available during the winter. So bats hibernate, survive the winter without eating, and those 4 species do that in caves. And this fungus kills bats in caves. It's a cave fungus. It's specifically adapted to live inside caves. The moving counterclockwise, the next 3 silver-haired, red, and hoary bats, these bats migrate south, and these bats live in trees, hoary and red bats in the foliage of trees, like you see here in the picture of the red bat hanging with its pup. It's 2 pups from the leaves. Silver-haired bats will often roost under loose bark. These bats don't go into caves, but they're highly migratory. They migrate long distance. So they're not heavily impacted by the fungus, but those are the species that are most heavily impacted by wind turbines. And finally, Big brown bats, that's the species that's least affected and seems to be doing best. A lot of people, when I talk about the 90% decline, will say, oh yeah, I've got tons of bats, they're all over, they're in my house every summer. These are always big brown bats. Big brown bats will spend the summer as well as the winter hibernating in houses and other buildings. They spend less time in caves and mines, and so they're not subjected to the fungus, and they're not long distance migrants, so they're not heavily affected by wind turbines. So my work and Bailey's work over the last couple of years has been focused in two directions: providing roosting boxes for them so that they have secure places to roost, where especially little brown bats, the one that's most heavily impacted by white nose syndrome, we want to provide them secure places to live, stress-free during the summer, so that they can remain in good condition, migrate in good condition, and have a maximal chance of resisting or developing immunity to the fungus. And then the other aspect of our research has been focused on mercury. Mercury is a contaminant, it's a neurotoxin. It's got a complex cycle that I won't talk about in any kind of detail. Comes in many forms, and some of those forms are not well studied. Human activities have released mercury and probably doubled the global pool of mercury. There's a great deal of debate about how much mercury is actually of human origin, but it could be as much as half. Methyl mercury is the form that is most toxic to bats and other animals. Mercury bioaccumulates and biomagnifies. That simply means if you start in the bottom left and you follow the arrows, small aquatic plants take up mercury. They become food for small animals, little zooplankton, that in turn are eaten by small fish, that are then in turn eaten by big fish, and the fish are then eaten by piscivores, animals that eat fish. It could be larger fish, it could be the human that's releasing the fish if he decided instead he was going to fry up that salmon for supper, osprey. And as you move through this food chain, mercury magnifies. It increases in concentration in each step of the food web. Most of the mercury or the mercury that's most dangerous in food webs, that gets into food webs most readily and is also most toxic, is methyl mercury. I'm going to switch back a little bit between total mercury and methyl mercury. For those with a science background, for those who study mercury, you can look at the slides and you can easily figure out whether I'm talking about total or methyl mercury. I'm not going to get bogged down for the general audience in those details, but they are on the slides for those who want to look at them. So one of the things I thought when I first arrived at the River Institute, where there were several people already studying mercury, I thought it'd be interesting to study mercury in bats. And the initial reaction among my colleagues was, don't bother, they feed atop a very short food chain. You probably won't see any high levels of mercury, certainly nothing biologically interesting. There's very little opportunity for biomagnification, because instead of having many links like in this food chain here, you have bugs feeding on decaying material in the bottom of the river. They turn into adults, and bats eat the adult insects. So you really just have two of these images and one of these connecting arrows, a very, very short food chain. So probably not interesting to look at bats, but I thought, well, I'm not convinced. Bats eat insects. They eat huge amounts of prey each night. They eat this red bat, one of the species I've studied previously, would eat somewhere between 50 and 100% of its body weight. And it's really hard to appreciate what that would even mean, but that would mean a grown adult my size that weighs 200 plus pounds eating well over 1,000 Big Macs. So they're really packing away the insect prey. They're eating large amounts. They're eating insects that live in aquatic sediments, the mud at the bottom of lakes and rivers and ponds, and that's where mercury tends to accumulate. Humans produce mercury emissions through various activities. It gets into those sediments. Bacteria turn it into that dangerous and bioaccumulative methylmercury form I talked about a few minutes ago. They live, bats that is, live more than 30 years, so they're exposed to mercury for a long time. It's very rare for a small mammal or a small animal, period, to live 30 years, to live to be 30 years old. So they're exposed for a long time. They live in urban areas where we often have industrial mercury issues. And finally, mercury accumulates in fur. I'm a behavioral ecologist, and the traditional method for studying mercury in fish is you catch the fish and you cut out a big chunk of its body and you put it in a blender. But I'm a behavioral ecologist, and when you put bats in blenders, their behavior is not very interesting anymore. And I wanted to be able to study something where I could sample mercury and then look at the effects of mercury levels on its behavior, and conversely understand how its behavioral choices influences how much mercury it ends up being exposed to. So my preliminary work about 20 years ago was some of the first work to look at mercury in bats. It was definitely the first work in Canada, and I very quickly went to a hibernation site like the one in the picture here. I sampled 4 different species, and I found all 4 species have bats that had mercury concentrations that reached or exceeded 10 parts per million. And that's not going to mean much to many of you, but 10 parts per million, that's the threshold that if a human woman had 10 parts per million mercury in her hair, and hair and fur is basically the same thing, we just name it differently when we're talking about non-human animals. That much mercury in a woman's hair, that woman would give birth to children with measurable learning deficits. So it's at the level known to cause learning difficulties in mammals. So the initial reaction to this work was, well, the sample size was small. Some of those 4 species, I only measured a few individuals. Must be some sort of analytical errors. You know, I mustn't have supervise my students closely enough. I'm sure someone at one point either said or thought, this is what happens when you let the biologist borrow the keys to the chemistry lab. You get crazy results. So we did some follow-up work with 2 different labs, so 2 different instruments, 4 different crews, and not only did we substantiate that bats regularly get to in excess of 10 ppm, we found even higher measurements in the 20 to 30 part per million range. So, and once again, to put in perspective, when a fish in its muscle has 2 ppm, we start getting worried about whether we're eating too much fish. And so we're talking about more than 10 times that value. The next part of the project was to look across North America from east to west. And so we sampled bats with help of a number of colleagues from British Columbia to Quebec. We relied on published data. Those are the ones in the orange bars from Little and colleagues, and then some American data from the northeastern US from Yates and colleagues. And what we found is Quebec and Ontario, where most of my data were from, were kind of in the middle, and mercury concentrations go up as you move towards the 2 coasts. And if you look at atmospheric deposition maps, basically where mercury falls out of the sky in rain or snow, you see that the red areas in this map are high deposition. So on both coasts, you've got lots of little red outlines of the coast. High mercury falling out of the sky there. The yellow is kind of medium amounts, so Quebec and Ontario right in the middle, and then the prairies are blue. That's the lowest deposition rate, the lowest amount of mercury or smallest amount of mercury coming out of the sky, along with rain and other precipitation. We also looked at fur and compared fur to other organs. And in fur, mercury concentrations are highest. They're lower in kidney and liver, and they're lowest in brain. But even, and this is just bats now, these are in little brown bats, but even in brain, we're getting mercury concentrations in brains of bats that are comparable to what you would get in the muscle of a big predatory fish like a pike or a walleye. So it really makes you wonder how bats are even managing to survive. Mercury is a concern because it's a neurotoxin, and neurotoxins affect brain behavior. They affect the ability for the brain to communicate with muscles and coordinate muscle movement. And bats have some pretty spectacular muscle movement that they have to undertake just in order to fly and hunt insects. So the scientists in the audience can look at p-values and the asterisks beside some of the numbers and the p-values. For the rest of you, this slide is just to remind you that we've also shown that mercury in fur, total mercury in fur, is correlated with mercury levels in several other tissues. So mercury in fur means the liver is processing and the kidneys processing mercury. It means really importantly that there's also mercury in the brain, which I just talked about. So to summarize what we know so far, 10 parts per million is not unusual in a bat. The pattern of variation in mercury in bat fur parallels the patterns of atmospheric deposition, and fur mercury concentration correlates with mercury in kidney and liver, and critically also brain. But what we still can't explain is why bats are so high relative to where they are in the food web. They look more like top carnivores, like river otters. And we also don't understand how bats manage to function with all this mercury in their bodies. And this is a slide that just shows where bats are compared to where they should be in the food web. So on the axis, if you follow the cursor, the arrow here, these are ranged from low trophic levels, so close to the base of the food chain. They eat either plants or insects, and moving to omnivores that eat insects, maybe also a bit of plants, maybe other things as well. And then at the far right, carnivores. And as bats are insectivores, they should be down here with this almost invisible thin blue line. But not only are they more like carnivores, they're right off the scale even to most typical carnivores. So they have insanely high mercury levels. We don't know why. It could be because they feed on aquatic prey, and mercury tends to contaminate aquatic ecosystems. They may feed in wetlands. Wetlands are natural filters, so wetlands have been filtering and removing contaminants, including mercury, for decades. That's one of the important functions of those wetlands. Maybe bats are largely feeding on wetland species of insects that are accumulating that mercury, or maybe bats have different ways of processing mercury that most other small mammals don't have. So our current activities involve trying to track bats to individual foraging sites using radio transmitters. You can see this thin line here in this bat. This is a radio transmitter antenna from a transmitter that this silver-haired bat is wearing. We're trying to document mercury concentrations in insects, but not just random insects caught in an insect trap. We're tracking bats to where they feed. We're figuring out what they eat, so we know we're measuring the right insects, the insects that the bats are actually eating. We're trying to document individual variation in the feeding patterns of different bats and see how that affects mercury accumulation. Lastly, we're looking at the molecular effects. So how does mercury influence bats at the molecular level? And with that, I'm going to let Bailey share her slides and bump my slides off your screens so that she can talk about some really exciting work that she is doing last summer and this summer with several other colleagues at the University of Ottawa. Are you good, Bailey?
Yep, can you hear me?
Yeah,
we can hear you.
Can you see my screen as well?
I'm just moving. We can see your slides, and I'm sending both live now.
Awesome. So hi everyone, thanks for joining today. I'm Bailey and I'm a master's student at the University of Ottawa. My master's research is focusing on two local bat species, Aptescus fuscus and Myotis lucifugus, which is the big brown bat and the little brown bat. And so my research involves looking at mercury levels in these two species. And I'm trying to do two things with the mercury data I obtained. So the first is trying to map out how mercury is moving throughout the bat's food web and trying to determine how the bats actually obtain such high levels of mercury. And then the second is to investigate how these high levels of mercury in the two species are affecting them on a molecular level. And so the molecular effects are what I'm going to be focusing on for this presentation. And so to start off, as Brian said, fur is a good way to measure mercury in bats. And so I've collected fur from a group of bats that Brian and I have collected together over the past few summers of me working at the River Institute, and also bats that Brian has collected himself over the past 2 decades. And so I have bats that were collected from the year 1999 all the way till this year, from a range of different areas, usually in close proximity to the St. Lawrence River, Cornwall area, surrounding areas, etc. And so I've measured the total mercury concentration in these bats' fur, and I have it plotted here on the graph. And so at the bottom here, I have the two species, so big browns here and little browns on the right. And on the side here, I have total mercury concentration in their fur in parts per million. And so each of these black dots represent an individual bat's total mercury concentration in parts per million. And then the bars here for each species represents the average or the mean total concentration of mercury. And so as you can see, the threshold Brian mentioned earlier of 10 parts per million, where you would start to see measurable learning deficits in humans, you can see that a lot of individual bats actually exceed this threshold, and that big brown bats specifically, their average sits right there at that threshold. And so that's pretty alarming. You can also see that big browns have a significantly greater concentration of mercury on average compared to little brown bats, which again has been shown in other studies. And so what I wanted to do with this mercury data was test the hypothesis that any differences between big browns and little browns in the way that they detoxify their bodies against mercury may contribute to this difference in mercury load between the big browns and little browns, making big browns maybe more tolerable to such high levels. And so before I get into the steps on how I tested that hypothesis, I'm just going to do a little bit of background information on molecular level metal detoxification. So the body has specific proteins whose jobs are to detoxify against heavy metals like mercury. And so the way that these different proteins do this varies depending on what the protein is, what it's detoxifying against, etc. And so very generally, once the cell is exposed to a heavy metal toxin, the cell needs to build these detox proteins so that it can detoxify against the metal. And so in order to build the protein, a detox gene has to be read, and in other words, transcribed in order to build these detox proteins. And so the way that the cell is signaled to start reading this gene is that the metal will enter the cell, and basically one protein will get activated, which activates another, and it's kind of just a chain reaction that eventually signals to the cell to read that detox gene and begins synthesizing the detox protein, which then goes and either eliminates the metal from the body and cell or binds to it and sequesters it, making its toxic effects. It kind of renders it untoxic to the body at that point. And so the two detox proteins I'm focusing on for my study is metallothioneins and metal regulatory transcription factor, or MTF1 for short. And so on this slide, I'm going to show you a visual that kind of explains how MTF1 and metallothionein work together to detoxify the body against mercury specifically. And so we have our cell on the slide here. This MTF1 in the orange circle is our detox protein. And so once mercury is exposed to the cell, MTF1 recognizes it and then binds to it. And so this MTF1 mercury complex is then able to move into the nucleus once this complex is formed. And it wants to move into the nucleus because that's where our detox genes are located, and that's where the synthesis of the detox proteins will begin. And so we want MTF1 to be able to move into the nucleus. So once this complex is formed, it's able to do so. So once it moves into the nucleus, it has to somehow signal to the cell to read this metallothionein gene in order to build the metallothionein protein. And so to do this, it has to bind to a section of the gene called the promoter region. And a promoter region basically just defines where the gene is going to start to be read and transcribed. And so in the case of MTF1 specifically, it has to bind to a section in the promoter region called metal response element, or MRE, which is represented by this green box here. So once MTF1 binds to the green box, it signals to the cell to read this gene, and it is transcribed, and you get your metallothionein protein, which then can go and bind and sequester mercury, rendering it non-toxic to the cell. And so big browns and little browns will go through this process when exposed to mercury in order to detoxify their body against it. But what I wanted to do was see if there were any differences in the way they do this between the two species that would somehow make big brown bats better able to tolerate the higher levels of mercury in their fur. And so the first thing I wanted to look at was the MTF1 protein. And so like any other protein, MTF1 is made up of these building blocks called amino acids. And the sequence of amino acids that make it up are what helps MTF1 bind to mercury to form this complex, and what help it bind to this MRE green box here. And so if there's any difference in the amino acids that make up this protein between big browns and little browns, it would potentially cause a difference to the way MTF is binding to mercury or to MRE, and therefore the metallothionein synthesis process. And so that's why I wanted to compare the MTF1 amino acid sequence between the two species. And so to do this, I basically used a set of online programs where you can search whatever protein you're interested in, and it'll give you this sequence of amino acids that make up that protein. And so I did that for big browns and little browns, and then pasted their sequences in another program that aligns them up with each other. And this program tells you how similar or how different the sequence of amino acids are from each other. And so what I'm showing here is the whole sequence of amino acids that make up MTF1 protein for little browns, whose sequence is the top line in every row, and then big browns, whose sequence is the bottom line in every row. And so I'm going to zoom in on a section of this so you can see it a little better. So again, little brown sequence is up here and big browns is right down here. And so when the program sees an amino acid, which are represented by single letters, where in places where they're the same, the program designates an asterisk underneath them. And that indicates to you that between the two species, these amino acids are the same in this position. And in positions where they're not the same, the program will leave that spot blank for example, here and here. And so if I go back to the whole sequence, you'll see that a lot more asterisks are present compared to blank spaces. And so what this basically means is that the amino acid sequence between big browns and little browns that make up this MTF1 protein are very much the same. And so it's likely that it behaves in the same way during the detoxification process. And so it's not likely a cause of big browns being better able to tolerate against the higher levels of mercury. One other interesting thing we found when we aligned these sequences is not only did we align the two bat species together, we also looked at the MTF1 protein sequence for 3 other mammals. So we did rats and mice because they're similar sized mammals as bats, and also humans. And what we found was that between the two bat species and humans, their MTF1 sequence of amino acids is very much the same, but not with the rodents. And so this shows that bats' MTF1 protein behaves in a more similar way to humans in this detoxification process than it does to other mammals their size. And so that was a pretty cool finding. And so the next thing we looked at was the number of metallothionein genes that each species has, along with the 3 other mammals. And so I wanted to look at this because if you think back to my animated slide where I had the metallothionein gene that had to be read in order to build the metallothionein protein to get the detoxification process started, if you have multiple copies of this gene, there's more genes to transcribe into metallothionein proteins, and therefore more metallothioneins being built and able to sequester more mercury. And so little browns have only 2 metallothionein genes, while big brown bats have 5. Mice and rats have similar numbers to the little browns, while humans and big browns are quite up there. So what this shows is that big browns compared to little browns have an expanded version of the metallothionein genes. And so they're likely to be more responsive to the exposure to high levels of mercury and better able to cope with them. And then the next thing I looked at was the number of MREs present in the promoter regions of these metallothionein genes. So again, thinking back to my animated slide where you had that green box on the gene where the complex had to bind in order to start transcription. If you have more copies, if you have more sequences of that green box, you'll have more spots for the complex to come in and bind and stimulate the synthesis of metallothioneins. And so, again, with higher numbers of MREs present, you'll be more likely to effectively respond to high levels of mercury. And so, again, little browns had much less than big brown bats did, where big browns had 8 and little browns only had 1 spot for this complex to come and bind and start transcription of metallothioneins. And so basically, to summarize my results, when we aligned the MTF1 protein between the 2 bat species, we found that their makeup is very much the same. And so this protein likely behaves in a very similar way for both bats during this process. and it likely isn't a cause for big browns being able to tolerate high levels of mercury compared to little browns. However, in support of my hypothesis in the previous slide, I showed that although little browns and big browns undergo this process in a very similar way, big browns kind of have an expanded version of it, given that they have more metallothionein genes and more MRE sequences where the transcription process is triggered. And so, as their name suggests, big brown bats have a greater body mass than little browns do, and so their feeding habits, in order to support this larger body mass, it may expose them to greater levels of mercury, and that may have played a role in them needing to develop this expanded version of the metallothionein detoxification network so that they're better able to tolerate the higher levels of mercury they're subjected to. And so in terms of what's next for my project, I'm going to continue looking at other molecular impacts of mercury in the lab. And while our results are very interesting and it's very novel, important research, the molecular impacts in bats, it still doesn't answer mine and Brian's original question of why do bats have such high levels of mercury even though they're sitting at the top of such a short food chain. And so some other steps we're taking is documenting mercury levels in insects, both terrestrial and aquatic emerging insects, to see how much they differ and if aquatic emerging insects really are subject to higher levels of methylmercury in the aquatic ecosystems. And we'll also be trying to determine which insects specifically make up the diet of each species of bats. And so once we determine the diet composition of both bats, we'll be able to combine that data with the mercury data in insects and the mercury data of bats and kind of map out the pathway mercury takes as it's bioaccumulating, and hopefully be able to determine how it reaches such high levels in bats at the top of this food web. And so that's all I have for today. I'd like to thank my supervisor, Jan Mennigan, for everything along the way so far, but also his expertise in molecular bio and ecotoxicology. And I'd also like to thank Brian Hickey for his expertise in everything bats and getting me involved in studying bats in the first place. And I'd also like to give photo credits to Stephanie Hildebrandt of the River Institute for taking so many fun pictures of all my research last summer, and they're throughout my whole presentation. And so, yeah, thank you, everybody. And so I'll stop sharing now.
Sure, yeah, thanks, Bailey. That was really good.
Thank you.
While we do questions, I am just going to put our upcoming River Institute events back up on the screen because we were pretty quick on that before, and we would love for everyone to know what's happening so they can take a look at that while we discuss. Up there, yeah, it's up there. Okay, so first question is from Kebby Brown. Hi, Brian and Bailey. Love the presentation and the project. I'm sure I speak on behalf of everyone when I say thank you for your work on a species that is so important for our ecosystem. I'm wondering if you've ever compared mercury levels in bats within Eastern Ontario, i.e., is there a significant variation in mercury levels on a bat living and eating right on the St. Lawrence versus elsewhere? Who would like to take that question?
I can have a crack at that, and if you want to add anything, Bailey, go ahead. So the challenge is that big brown bats are more urban in general, and little brown bats are more rural. So we don't have a lot of little brown bats from right within the city of Cornwall. And we have lots of big brown bats from right in the city. Now, so bats in Cornwall are more contaminated than bats outside of Cornwall, but we don't know if it's because they're in Cornwall, where there's more industrial mercury in the sediments, or if it's because they're big brown bats, and big brown bats are more contaminated. Now, with Bailey's work, plus my previous work, I think we might finally have enough of a sample size to tease those two autocorrelated things, tease them apart. But we haven't done that yet, or at least I haven't done that yet. Have you done that yet, Bailey?
No, not yet. I have mercury data from bats in surrounding areas, so Mooresburg, Somerstown area, further that way, but I haven't run any statistical analysis to determine how different they really are.
And we also haven't merged, Kebby, some of my older data with Bailey's new data, so that's a great project for us this winter. Great to hear from you, and thanks for suggesting that. For the rest of our listeners, Kebby, I think, might have been the person climbing that ladder inspecting the bat house. Kebby has been part of our crew in the past as well.
Perfect. All right, thanks, you two. The next question. Are you planning to do stable isotope analysis on your bat samples to differentiate trophic levels?
I believe I'll answer that one because I think that's for me. But yes, I just filled out a form since the labs at U Ottawa are open again. I just filled out my form with all my samples on it, so I'll be running it on the bat fur and the insects in order to determine which insects are making up what percentage of the bat's diet.
Perfect. Good. Okay. Brian, this question is for, well, actually for either one of you. The overall trends for little brown bats. I know when we were in the office, I would get to hear anecdotally, you know, when a bat was brought in or when you were going out to set up a new house somewhere, and just more generally, if you could speak to the trends for the little brown bats.
And you're referring to population trends as opposed to mercury trends?
Yes.
Yeah. So we so although the declines in caves have been spectacularly scary, the two pictures I showed, we still are finding little brown bats locally. In fact, the other part of the project, deploying bat houses and trying to give them safe roosting places, I've lost track now, 5 or 6 bat houses with quite large colonies that seem to be stable. So there are a few populations hanging on locally. I'm hoping what I'm seeing is the beginning of resistant populations, and I'm hoping in the not too distant future, we start to see bats moving back into some of those hibernation sites. So I'm cautiously optimistic that little brown bats may have already reached the bottom and are, if not climbing, at least holding stable at these lower levels. The 3 species that I didn't have the photos for, that I just listed on the side panel of the 8 species of bats in Ontario slide, those, some of those may be in real dire straits. They may be gone or functionally extinct and not able to recover. They were already rare before the fungus, so I'm more worried about those. I think little brown bats are going to survive.
Good. Okay. So the next question is, are there any differences in mercury levels between male and female bats? Females sometimes transfer their body burden to their young. Who'd like to take that one? Bailey?
Did you want me to start, Brian?
Sure.
Okay, I have compared the two, and with the Mercury data I have, I didn't see a significant difference between male and female. The only significant difference I found was between age, and so younger bats having much less mercury than adults did, given that they're exposed to it for a much longer period of time. But for sex, I haven't seen anything, but I think other studies have found differences, right, Brian?
I think so. So I think our sample sizes and our numbers of studies is sufficiently low that I'm not sure. I'm not sure there's a consensus on that issue. In my earlier work, some of my earlier work, there were no differences, but as Bailey said, in other studies, there have been differences. Often the challenge is our sample size is so dominated by females, because when you go to if you find a colony in the summertime, it's typically a maternity colony of females and their offspring, and the males are elsewhere, and they're much more nomadic and hard to find. So the sample sizes are really skewed. So we need to get more data from male bats. But that is a common thing you see, is mercury moving out of the adult female's body and moving into the fetus that's developing.
Great, thank you both. We have a couple minutes left, and I was thinking, it's not a science question, but it's a anecdotal story that I love about the River Institute and the role that the Institute plays in our community and with the students that we have through our doors. So Bailey, you have told the story before, but I'd love it if you could tell everyone again about how you decided to do your master's program.
Okay. So I started doing my undergrad at U Ottawa in biology in order to get prerequisites so that I could go to veterinarian school afterward. And then about halfway through my undergrad, I got a summer job at the River Institute working as Brian's research assistant. And I've always loved science, but ever since I started working with Brian and he started helping me be able to apply all the things I learned in the classroom to real life situations and the field work we were doing. And so it never really meant anything to me learning it in the classroom until I was able to apply it to the real world and the real work that was being done by scientists. And so, and also he showed me all summer how cool bats are. And so I decided that I liked the research aspect to science more than I think I would have liked vet school. And so I decided to do my master's after that.
Amazing. That's great. Brian, did you have anything to add to Bailey's story?
No, well, I think yeah, it just took me a minute to unmute myself there. No, I mean, other than it's been great to work with Bailey and through Bailey be connected with her other supervisor at Ottawa U, because we're expanding this work into molecular biology, which is not my expertise. And I think the real strength of this work is that we're going to be able to look at things from an ecosystem and a predator-prey level right down to the molecular effects level. And I think many of these questions about differences between males and females and trends between urban and rural bats, these will all be answered in the course of this collaboration, and we'll have a more complete understanding of why bats are so different than all the other animals in terms of how quickly and how much mercury they accumulate.
Wonderful. That sounds good. And thank you both for your great presentations. Bats are definitely one of the coolest topics out there in the natural world. People have very strong opinions on them. We will wrap it up for tonight. So thank you everyone who tuned in to watch the presentation. Our next Science and Nature Untapped is August 5th with Barbara King from Watersheds Canada. And thank you also to your TV for broadcasting the River Institute's Science and Nature Untapped for local cable viewers. And if you have any questions, always send us an email or send us a direct message on our social media channels. We'd be happy to connect. Have a wonderful evening, everyone.